Good morning. I'm Tom Cooper, and it is my pleasure to be moderator of the poster lightning round. And so we've had over 40 posters submitted. And so just to remind you, we have poster session at 12:30. That'll be in the main room. So I encourage you to see all the posters. There were 9 posters that were selected for these presentations. These are 5-minute presentations. We will have time for a few questions after each presentation. And these posters were selected, and of course it was difficult to select which posters to present, but they were selected based on the diversity of what is going on in the myotonic dystrophy field and all the different labs that are working in myotonic dystrophy field. So we will start off. I'd like to remind the speakers, please keep to 5 minutes. Just in terms of a fairness for those who did keep to 5 minutes. And also it will give us time so you can have questions after your talk. All right, so let's get started. The first speaker is Johanna Hamel from the University of Rochester from the Thornton Group. And I also note the poster number, so you can write it down if you'd like to hear more about it and go visit the poster, because these individuals will also be at their posters during the poster session. So it's poster number 45.
Go forward this way. Okay. Yeah, good morning. I will be presenting on the ongoing and actively enrolling study, REACH-DM Remote Assessments and Genetic Analysis in Patients with Myotonic Dystrophy Type 1. The goal of this preliminary first analysis of the first cohort is to further assess the feasibility of measuring multisystemic manifestations of DM1 remotely, and also to assess the test-retest reliability of measures done remotely. The workflow is as follows. We recruit through the National Registry and eventually the MDF registry. A toolkit is shipped out to participants to their homes. We remotely consent participants. Then the participants fill out questionnaires on their tablet that we provide for them. Then a remote study visit takes place, which takes about 2 to 3 hours. Then participants have their blood collected in their home environment or in their home community and return it to us. And then they return the toolkit to us, and we recycle and clean the toolkit and ship it out to the next participant. Study procedures are depicted here. The 9-hole peg test, grip strength, forced vital capacity, sitting and supine, tongue strength, sniff intranasal pressure or SNP, tug timed up and go, video hand opening time, pinch strength, an EKG is done, and they perform the 10-meter walk run test if their home environment allows to do so. And here on this slide, you see how many people were able to complete these assessments in their home environment out of 32 for each procedure listed. You see that that nearly all participants were able to complete all assessments. I would just like to highlight the blood collections. We received blood from 29 participants already. And I would also highlight the 10-meter walk run test, as we were able to do this test in the home environment in 24 people. This slide shows the average results of each test. And I just want to highlight here that we had a broad spectrum of disease severity in these participants. For example, the grip, you can see the first and third quartile, 7.5 and 22 kilograms. We had people that had respiratory weakness. As you see, the FVC percent predicted, the average was 80% sitting, but with a standard deviation of 23. And then the 10-meter walk run test also had a wide range of speeds. Then we went on and tested test-retest reliability by doing remote study visits back-to-back within 30 days. And here, this graph, similar graphs will show up on the next couple slides. So I'm just going to orient you. So this is the results for the 10-meter walk run test. On the y-axis, you see the seconds. On the x-axis, you see individual subjects listed, and each of them has 2 consecutive tests listed. And you see that both tests done within a month time frame were close to each other. With an ICC intraclass correlation coefficient of 0.99, which is excellent. And then here you see hand grip, 3 trials averaged versus the best one picked. And this ICC for both was 0.99. Also, you see that the 2 test measurements are close to each other. Here you see the retest reliability for the 9-hole peg test done remotely. And again, the ICC is excellent at 0.98, and the 2 consecutive tests fall close to each other in terms of performance. Here I highlight the forced vital capacity, the breathing test done supine in the home environment on the floor or in, wherever people would safely lie down. And the intraclass correlation coefficient, again, is excellent at 0.97 on those 2 consecutive measurements. The tongue strength also is a little bit more variable, but still excellent with an ICC of 0.94. The timed up and go is more variable in the on-site research study setting as well, and maybe a little bit more variable in people that are more severely affected. But again, the ICC is acceptable. And then the most variable symptom was or assessment was the video hand opening time. And here I would just like to comment on that people were allowed to be on anti-myotonia medications, that they took variable to the timing of the remote study visit, as well as the resting time preceding the test was sometimes a little hard to enforce. So we're making some modifications to that. And then graphs I didn't show, but here is the retest reliability of procedures done, which for the most part you see pinch, FEC sitting, SNP, buccal strength is acceptable, with ICCs mostly greater than 0.9. So the conclusion is that remote assessments of multisystemic manifestations of DM1 are feasible. The retest reliability so far appears acceptable. And this approach may offer a broader engagement of the DM1 community and studies of larger cohorts, which is what we envision. The VHOT is more variable, but again, I mentioned the limitations in this first cohort. And with that, I would like to close and thank particularly all participants and their families and friends, and the team in Rochester.
A few questions. Microphone, please.
Sorry, I was just curious because you have the data for in-clinic. The video hand opening time has variability too, even when it's supervised and seated. I'm just wondering what the comparison of the standard deviation is for that.
Yeah, I don't have that data. I think we have that in Kate has that, and John raises his hand. Maybe he has it. But we have done on-site, or we're starting to do on-site comparisons in that cohort. And we have a handful of patients who already came by and we're testing that. I think it's better, but the resting times, as you know, usually 5 minutes. And then in the Marina trial, we do it back to back. So I think it's better, but I don't have that data here. And we're collecting our own data on this. Yep.
Thanks, Johanna. I thought that was a great presentation. And as you know, I think it's a really important thing to do. So thanks for developing this. I was curious about the dropout of some of the assessments, because these were all monitored visits, I think. So why did you not have a full 32? I understand for the 10-meter walk run. And my question about that is the implications of, can we move to even having non-monitored assessments? Can we ask them to do multiple assessments, or is that just going to get even noisier?
Yeah, no. So the dropout wasn't, I know it maybe looked like a dropout, but it wasn't a dropout because the big goal is to enroll a large cohort. And we have a sub-study, which includes participants who are willing and able, have the time and the capacity to do another remote study visit within 30 days. So it's not that the other ones aren't capable or so, but just they sign up front to do the repeat study visit. So we are still enrolling for the retest reliability. So this is just a preliminary look at the initial data. So it's not technically a dropout. It's just a sub-study. Yeah. And then unsupervised procedures, yes, definitely. I mean, it's been done for the hand grip before, and that worked really well, looking at that initial data. And there are certainly some assessments that people can do unsupervised and a video record. I know you're doing work towards in that direction. Definitely. I think it needs some assessments lend to that more than others. And I'm gaining experience where people need more guidance, but hand grip, pinch strength, those kind of things are probably amendable. For example, the breathing tests, maybe not so much. Yeah.
Great. Thank you very much. All right, next, the second speaker will be Janelle Peterson from my lab, and this will be poster number 13.
Okay. Perfect. Is it okay if I reset the timer? Yeah, cool. Okay. Awesome. Hello, everyone. Thank you so much, Tom, and thank you so much to the organizers for this opportunity to speak. I'm really excited to share with you the latest findings that we have on the role of muscle blind and smooth muscle function in DM1 GI pathologies. So as many of you may be aware, a very large proportion of those affected by DM experience GI symptoms that affect any and all parts of the GI tract, from trouble swallowing at the esophagus to fecal incontinence at the anal sphincter. So dysfunction of the GI tract not only impacts your day-to-day life and well-being, but it also really impacts your health. We all need proper GI motility in order to consume and digest food, without aspirating it, to uptake water and nutrients from that food, as well as to expel our waste when we want to. So this is all accomplished through the coordinated contractions of the two muscle layers that line our GI tract. We have the circular layer, which is controlling the gut lumen diameter, as well as the longitudinal layer, which is controlling the gut length. When functioning correctly, food should be churned and propelled forward down the GI tract. So two factors play a really large role in whether these two muscle layers are contracting properly. We have neurogenic factors like the enteric nervous system, which are inputting signals for that tissue to contract. However, we also need the proper response by those muscle cells. So our lab decided to focus on whether known mechanisms of DM, in this case, the loss of muscle blind 1 and 2, within the smooth muscle cells alone can cause GI dysmotility. To accomplish this, we generated a mouse model with a tamoxifen-inducible smooth muscle-specific Cree with flox MBNL1 and floxinol MBNL2 alleles. So our experimental mice that are losing muscle blind specifically in the smooth muscle are denoted as SMO Cree DKO throughout this presentation. And all of the mice, including our controls in the following experiments, received 10 days of tamoxifen treatments and had a 4-week washout period prior to phenotyping. So to first assess whether muscle blind loss is actually affecting GI motility physiologically, we used a dye-based assay. So we placed a thick C dye conjugated to a very large dextran molecule into the stomach of mice by gavage. And after 25 minutes of allowing that dye to move through the small intestine, we can dissect out the GI tract, section the small intestine, and then rinse out each one of those sections, allowing us to see how much of the dye was present in each segment. So we found that in our control mice, shown here in black and gray, on average had the greatest amount of dye, or the dye bolus, present in segment 7, represented here by the larger circles. However, in our smooth muscle Cree double knockout animals, we see a shift where now the majority of that dye is in segment 5, and this is indicative of delays in small intestine motility. So upon dissection of our mice, we observed that the small intestine was significantly reduced despite mouse weight and mouse length not being any different between groups. And surprisingly, upon histological evaluation, we were surprised to find that these smooth muscle Cree double knockout animals actually had increased muscle and mucosa thickness in some regions. We also did not see any change in cell death by tunnel, and this indicated to us that atrophy of the smooth muscle was not occurring. So now this is suggesting that there is an intrinsic smooth muscle cell defect occurring. So looking at the transcript level, we did identify previously known muscle-blind sensitive changes, such as changes in NFIX, but we also identified new smooth muscle specific ones. So for instance, we found alternative splicing in myosin light chain phosphatase subunits, as well as in tropomyosin 1. So finally, we assessed contractile force of living mouse GI segments in an organ bath. And in this video, we have a living segment of a small intestine that's connected to a force transducer by black thread. And below is the data that we're actually collecting. So as the tissue is contracting, the force is increasing and we have peaks. And when that tissue relaxes, we have valleys in the data. Overall, we found that our smooth muscle Cree double knockout mice have decreased contractile activity. So I hope today, within my 5 minutes, I've convinced you that the loss of muscle blind in the smooth muscle alone contributes to GI dysmotility. And while I focused on the upper GI tract today, the lower GI tract shares similar phenotypes. And I'm really happy to discuss these findings at my poster later this afternoon. With that, I'd like to thank all the individuals who have gotten me this far, as well as my funding resources. And to see more of the Cooper Labs work, please check out our postdoc Larissa's poster as well. And I'm happy to take any questions. Thank you.
Questions? Great talk.
Thank you.
Is the expectation that you have to have fairly significant sequestration? Sorry, thanks. Because of the double knockouts where you're seeing the strong effects, did you check in an MBNL1 or an MBNL2?
Oh, alone?
Yeah.
We opted to just go directly for the double knockout, since in mouse models, it seems like that contributes to the most significant phenotype. And the GI tract is, as we all probably are aware, is very variable. So there's a lot of stressors that can affect it. So we really wanted to make sure we're actually able to capture the phenotype. So that's why we opted that way. We are thinking about doing some single flox alleles as well, just to see, but it was a good place to start.
And then in terms of maybe a CUG model?
Oh, we would love for that to happen, hopefully coming down the pipeline.
So, this. Okay. So this is really exciting to have a model of GI motility, because this is such a huge problem for our patients. Do you think the model that you have of looking at the contractility would be something that you could use to look at pharmacological effects like opiates and anticholinergics?
Absolutely. Absolutely. It's the organ bath experiments are really awesome because you're able to put anything into that organ bath to see how the tissue reacts to it. So we could look at different gut hormones. We could look at acetylcholine agonists, as well as others. So that's in the plan in the future directions.
Just a clarification.
Yeah.
The dextran is put into the stomach or into the small intestine?
The stomach. So the gavage goes directly into the stomach. Yeah.
Right. So it could be either a problem at the gastric outlet or in the small intestine.
Yes. So after the 25 minutes, if there potentially was some gastroparesis occurring, we would have most likely seen more dye in the stomach. That's one of the segments we include in the washouts. And when I've done the statistical analysis, it looks to be emptying out properly, but we could do some other assays where we shorten the amount of time to see how much is actually released.
I was just asking because of that genome-wide association study that showed the polymorphism near MBNL1 was associated with gastric outlet obstruction in infants, right? Yeah.
Absolutely. Yeah. I was anticipating that we would see a delay in gastric emptying, but not particularly here. There might be other factors that are contributing to that. Maybe the enteric nervous system that we're not capturing here.
Real quick, are you able to, when you dissect out the intestine, look for changes in absorption? So can you take those sections and look to see if changes in osmotic,
absolutely
salts and whatnot?
Yeah. So there's Using chamber studies that you can do where you basically take out the living segment, you flay it open, and then you can see how FITC moves across that tissue. In this case, we were just focused on motility. So that's why our FITC dye was conjugated to the large dextran molecule. It's impermeable to the tissue, but it is very much possible.
Great. Thank you very much. All right, our next speaker is Claudia Lennon from the RNA Institute at SUNY in Albany from Andy Berkman's lab. And this is poster number 29.
Hello, everyone. Okay. So I am a bioinformatics support specialist at the University of Albany's RNA Institute, Albany, New York. And the title of my poster and talk is Individual Transcriptomic Response to Strength Training for Patients with Myotonic Dystrophy Type 1. So the content of this talk is from a recently published paper of the same name. It's a collaboration between the Berglund Lab and the Duchenne Lab in Quebec, Canada. And then this study, the data came from a previously conducted clinical study in the Saguenay-Lac-Saint John region of Quebec, Canada, where male DM1 patients participated in a 12-week strength training program, specifically targeting the quadriceps muscle of the upper leg, with muscle biopsies taken before and after the training program. All of the DM1 participants saw clinical improvement, significant improvement in their strength and physical capacity following the program, and that clinical data has been published. But then it led to a further research question was if we saw improvement at the clinical level for all DM1 patients, were there also improvements seen at the molecular level? To answer this question, RNA sequencing was performed on the pre and post-exercise muscle biopsy samples for all DM1 participants, as well as for control individuals who don't have DM1 and did not participate in the program. And then today, the content of my talk will be going over the results for differential gene expression and alternative splicing analysis on that data. So to begin, I'll be going over some results we saw for the alternative splicing analysis after strength training. We observed widespread effects in skipped exon alternative splicing across the individuals with every DM1 participant, except for the one identified as individual 907, having a majority of skipped exon events that were misspliced prior to exercise, rescued after strength training. Rescue in this study we defined as a 10 to 110% shift in percent spliced in values to control levels. We found that many rescued events were unique to the individual. A large number was shared between or amongst two individuals of the study, but we found that as the number of individuals increased, the number of shared events drastically decreased, and no single event was found to be shared amongst all of the DM1 participants. We also analyzed splicing dysregulation. We did this by calculating a splicing dysregulation score following strength training. And the exact calculation is further discussed at my poster, but here in this figure, you can see a decrease in the score indicating improvement in splicing dysregulation following strength training. That was observed for all individuals, even though only slightly for individual 907. Key finding from this analysis was that we also calculated a score in a grouped version as well as per individual. And this group version seemed to mask the significant changes seen across multiple individuals of the study. So for example, here with individual 523, a robust score change not seen in the grouped version. So it seemed that the grouped analysis did not accurately represent what was being seen at the individual level per patient. We also analyzed potential changes in gene expression, and changes were seen, but with considerably more variability amongst the individuals than with the splicing analysis. A large range in the number of genes rescued after training was observed, but the global changes in differential gene expression across individuals did correlate significantly with their clinical improvements measured. So their percent of differentially expressed genes rescued correlating to their average percent change in 1 repetition maximum measurements from their lower limb exercises. Several of the individuals saw improvement in differential gene expression dysregulation at varying levels after training. And again, we saw that a grouped analysis masked what was seen for the individual scoring, again, highlighting a change for the individual 523 versus the all group. This masking effect was further investigated in this study by taking previously published cycling training data from another DM1 exercise program. This study used a grouped analysis to determine the effects transcriptomically between the DM1 participants pre and post exercise. And they For this study, we reanalyzed that data at the individual level and saw significant changes for individuals following the cycling training study that was not previously included in the first cycling training study. So for conclusions and future directions, our studies suggest widespread transcriptomic effects of this 12-week strength training program on DM1 patients at the individual level, adding to the previously confirmed clinical benefits. The transcriptomic response had varied amongst the DM1 individuals, with significant changes not reflected when a grouped analysis was performed, suggesting a need for future studies to consider investigating DM1 at the individual level. And exciting future work includes transcriptomic analysis of a female DM1 cohort who recently completed the same 12-week strength training program. And you will be hearing about that clinical program next. So to end, I'll do my acknowledgements. So I want to acknowledge the partnership between the Berglund Lab and the Duchenne Lab. All the individuals bolded are those involved in this published study. And a special thank you to the Berglund Lab past members, specifically Emily Davey, who is the first author on this paper. I'd like to thank also our other collaborators, the funding agencies and universities involved in this project. And a big thank you to all the participants of the exercise study, their families and caregivers. Everybody that is starred are also presenting a poster here at the MDF conference. So please be sure to check out their work. And in my poster will be number 29. So thank you for listening.
Excellent talk. What are the proposed mechanisms and updates of improvements in the transcriptional changes?
Right. Yeah, so this study did not go into depth about any specific mechanism, but it definitely seems to be a complex response, especially since the individual scores and the individual levels of rescue seem to vary quite broadly. All the individuals improved clinically, but at the transcriptomic level, it seems to be perhaps related. Some of the suggestions in the study included perhaps related to DMPK expression or MBNL enrichment, certain aspects like that.
Thank you for your talk. Did the exercise study include control myotonic dystrophy patients who did not undergo the exercise training to assess sort of what the baseline level of stability is for missplicing and differential gene expression?
Yeah, good question. No, it did not. It included just the DM1 participants that underwent the training program and then controls without DM1 that did not undergo the training. So for this study, it was really important to get recruitment and participation within it. So it was a limitation of the study, just the small sample size that we had for DM1 participants. Yeah.
What do you think participant number 907?
Yeah, certainly an interesting response for it. So for that participant, again, amongst all the participants, they had one within DM1 it being heterogeneous, but them as individuals themselves had a lot of differences. So we saw differences between CTG repeat length, age, BMI that was calculated for each of the individuals. So all of that included, it's still unclear exactly why 907 compared to other individuals had some differences. We proposed some in the discussion section of the study, but still unclear. And hopefully when we look through the female cohort transcriptomically, we can see something that can help us figure out exactly what the individual response is.
Great. Thank you very much. All right, next speaker is Laura Girard-Cote from the University of Quebec, from Cynthia Gagnon's lab, and this is poster number 40.
So thank you for receiving me today. So I'm a physical therapist and a master's student. So I present to you briefly my master's project. So as we know, myotonic dystrophy type 1 is a prevalent neuromuscular disorder, and it's even more prevalent in the region of Saguenay-Lac-Saint-Jean, making it a great place to study the disease. And as a reminder, this is a multisystemic disease, and lower limb muscle weakness is a cardinal symptom in the disease. So to overcome these impairment, it is interesting to making a strength training program. So it has been studied before on a cohort of men, like Claudia just said before me. And it has shown various positive effects on the clinical and the fundamental side of the project. However, there is clinical differences between men and women. So in our study, we aim to assess the impact of a 12-week lower limb supervised resistance training program in women with DM1 on the lower limb strength, physical function, neuropsychological health, muscle histology, and metabolism. So the DM1 woman went twice a week training at the University of Quebec while 12 weeks. Each participants were paired and supervised by myself or a kinesiologist. There were 5 exercises at the program, such as the squat, leg press, knee extension, hip abduction, and ankle plantar flexion. Each participant did 3 series of 6 to 8 repetition maximum of each exercise. So the study had 5 time points of measurement, such as before, during, after the training program, and also 6 months and 9 months after the beginning. Muscle biopsies were done before and after the training. Also, we done muscle strength assessment, physical function, falls, pain, and neuropsychological assessment at all time points. Statistical analysis were done with NPAR-D, non-parametric repeated ANOVA. The first comparisons were done between T0, T1, and T2 to see the effect of the intervention. And the 2 last comparisons were done between T0, T2, T3, and also T2 and T4 to see the maintenance of the gain from the intervention. So we had 11 women who completed the training program with juvenile, adult, and late onset phenotype. The training attendance rate was of 98.5%. As the 1 repetition maximum strength, we assess each exercise at the training program. We can see on each graphic that there is a significant improvement for each exercise after the training program. But there was only for the squats and the ankle plantar flexion, there was a maintenance of the 1 RM straight up to month 9. So for the quantified muscle testing, there was only a significant gains for after the training program for the knee extensors and the hip extensors. And the maintenance of the gains were maintained for the hip extensors only up to month 9. For the functional assessment, there was only at the 30-second sit to stand that there was a tendency of improvement after the training program. For the self-report questionnaire and the neuropsychological health, there was a significant improvement after the training program for the lower extremity functional scale, the pain interference in the brief pain inventory, the apathy score, and also for the anxiety score at the hospital anxiety and depression scale. And all these gains were maintained up to month 9. For the muscle biopsies, there was a significant improvement of the mitochondrial respiration after the training program that rescued to control levels. As for the other histomorphological feature, they are currently under analysis, but we have some preliminary result on the mean minimal ferrite diameter of the myofibers. So there is a tendency of increase after the training program. As a conclusion, the strength training program seems to be a positive therapeutic approach in women with DM1. As I just said, some results are still under analysis, particularly for the fundamental side. But to date, there is still a lack of information for the healthcare professional, and it is for that reason that we are currently doing a video of knowledge transfer for the clinicians and the person with DM1. So I would like to thank the participant and all the people involved with the study. Thank you.
Presenters are doing a great job staying on time. Thank you. Questions? Actually, I was quite impressed that you have 98% people.
Yes.
I mean, that's doing better than, I don't want to say anything. So that's impressive. How about well-being? Was there, and you did have some measurements on anxiety.
Yes.
Did you get a sense of a change in well-being and a feeling of well-being?
But we don't have objective changes about that unless all the questionnaire that we did in the study. But we made a focus group after the intervention. Like we asked the participants how they thought there was the training program, how did they like it, and we had so much positive and great comments about the training program. It was like awesome to see that. Seriously, you should have been there. I didn't have the time to present these results, but they're so beautiful.
Thank you for your talk. That was really cool. One of the things I was wondering is, did you ever collect data on what the patient's previous history was with like any strength training programs? Like, were they all beginners, or did they have any other mode of exercise prior to the study? Just because some of these numbers are super impressive just in general. And so that's something that we're really interested in.
Yeah, at the beginning of the training program, we asked if they did before strength training or any training. I think only one of them was training before, but for the others, they didn't train at all in their life.
That was great. Do you have any overall conclusion about the response in men versus women? Because the stimulus is about the same but across the two studies. Is that true?
Yes, yes.
So what's the difference in response?
We're currently analyzing that because I just finished the training program. So there is differences. If I remember well, I think that in the men, they had the positive impact on the fatigue and daytime sleepiness. And we didn't have that thing with the woman. And also with the woman, we had positive effect on anxiety that they didn't have on men. But for the strength, it was almost the same. I don't know the percentage, I didn't compare it, but there's a positive effect for the woman and the man.
All right, thank you very much.
Thank you.
Our next speaker is Preeti Kumari from Mass General and Thurman Wheeler's lab. This is poster number 39.
Thank you so much. Good morning, everyone. My topic is presentation is extracellular RNA splice event in cerebrospinal fluid as a candidate biomarker of myotonic dystrophy type 1. So with recent development and progress, biomarker act as an emerging tool for the drug discovery and development. And FDA categorized these biomarker into the following categories like diagnostic biomarker, response biomarker, monitoring biomarker, predictive biomarker, and prognostic biomarker. As we all know that the myotonic dystrophy is a multisystemic disorder that involves central nervous system. And previous study reported the accumulation of RNA foci in cortical neurons, and several study reported the missplicing of alternative splicing in DM1 brain and mouse models. So However, biomarker of molecular disease activity in DM1 CNS are unavailable. So previous study reported that these extracellular vesicles carries the biomarker for cancer. So we came for the extracellular vesicles. So these extracellular vesicles are membrane encased particle, and they are form of extracellular communication between the cells. These extracellular vesicles carries messenger RNAs, non-coding RNAs, and microRNAs, collectively termed as the xRNAs. On the basis of origin and the size, these extracellular vesicles categorize into the two categories, exosomes and microvesicles. Exosomes are 30 to 100 nanometer in diameter, and they are formed by the multivesicular bodies that is shown here in yellow. And microvesicles are 100 to 1000 nanometer in diameter, and they are formed by the exocytic budding of the plasma membrane. From the recent studies, these extracellular vesicle has several applications in drug delivery and also in the diagnostic as a biomarker, because they are carrying the disease-specific cargos. These extracellular vesicles are released from the variety of central nervous system cells. For example, neurons, microglia, oligodendrocytes, and astrocytes. And these extracellular vesicles participate into the overall regulation of neurological activities. So we hypothesize that the cerebrospinal fluid contains a representative transcriptome that is altered in DM1 and quantifiable in EVs. For that, we collected the cerebrospinal fluid from the unaffected control and the DM1 patients, and we performed the isolation of CSF cells and isolation of extracellular vesicles separately. For CSF cells, we performed the low-speed centrifugation, and for isolation of extracellular vesicles, we performed the size exclusion chromatography or ultracentrifuge. And after that, we performed the droplet digital PCR for the alternative splicing analysis and the gene expression analysis. We characterize the presence of these extracellular vesicles in CSF, and we compared with the extracellular vesicle present into the urine and serum. So the CSF EVs are categorized under the microvesicle categories because they are around 160 to 200 nanometer in diameter, and the concentration of CSF EVs is lower in comparatively to the urine and serum. We performed the gene expression analysis of the genes that is responsible for DM1 and DM2. So the total copies per microliter of the both genes are higher in cells, but the gene expression data is normalized gene expression of DMPK and CNBP is higher in xRNA. So that this finding concluded that the extracellular RNA, the source of the EVs into the CSF is more prominent to CUG pathology because it contains more expressed DMPK and CNBP. After that, we performed the alternative splicing of the already known transcript that are misspliced into the DM1 brain and the urine EVs. So here, percentage exon inclusion of 23 is higher in DM1 xRNA and comparatively to the unaffected control, and there is no change reported into the DM1 CSF cells. Also, the NUMA1 and COR2 and MBNL2 is misregulated into the CSF xRNA, but there is no change has been reported into the CSF cells. So from this study, we concluded that the quantification of CNS-derived alternative splice product in CSF xRNA is feasible. GOLGA4 is a candidate biomarker of early CNS involvement in DM1, while NUMA1 and COR2 and MBNL2 splice events are candidate biomarker of disease severity. In future, we wanted to determine the relationship between CSF xRNA biomarkers and clinical indicator of CNS involvement in DM1. We also wanted to perform the transcriptomic analysis of to identify new misspliced transcript in central nervous system. Thank you.
Thank you. Questions?
I'll just talk loud. So great talk. Did I understand correctly that there's missplicing in the exosomes for the transcripts, but not
in the cells?
But not in the cells.
Yeah.
And do you have thoughts on maybe that differential, why that happens in terms of mechanism?
Because the cells, they are basically leukocytes and the blood cells, and they don't have they don't express the expression. Yeah.
Thank you. That's really great. It's very exciting. Do you have any sense of the cell of origin of the exosomes or how you might even begin to address that?
Right now, we don't know what is the origin of the cells, but we can, in future, we can find out, because these extracellular vesicle has the receptors and that is specific to the CNS cells. So we can find out what kind of the origin of the cells, exosomes.
All right, very nice. One more question.
One more. So just curious to know, have you been able to, I'm over here. Hi. Just curious to know if you're able to correlate anything with repeat length in the biomarkers?
We don't have enough DM1 sample right now, but in future we will do the more when we have the enough data, then we will correlate.
Very nice. Thank you. All right. Next speaker is Emily Davey from University of Florida in Gainesville from Eric Wong's lab. This is poster number 43.
Hi, everyone. Thank you for this opportunity to talk here today. And I will be telling you about work we've done on investigating global and cell type specific transcriptomic dysregulation in the DM1 brain. So as many of you all are familiar with, peripheral symptoms are commonly studied in DM1. However, CNS symptoms are known to be a significant burden to patients with DM1. Some of these symptoms include things such as memory deficits, hypersomnia, apathy, to name a few. And neuroimaging and neurocognitive studies support there's a widespread CNS involvement in the DM1 CNS. However, our knowledge of molecular mechanisms of this involvement are limited. So previous work from our lab has shown that splicing dysregulation is widespread across the DM1 frontal cortex. As you can see here, showing the unaffected mean and DM1 mean of percent spliced in, or psi values, with the significant events shown in blue. You can see there's widespread missplicing in the frontal cortex. And as shown in the heat map here, you can see that there is a wide range of severity between DM1 individuals and between different events. However, what we don't know is whether splicing dysregulation is localized to certain brain regions or whether it's widespread across many brain regions. And so it's as though we're trying to get the whole big picture of transcriptomic dysregulation in the DM1 CNS, when really in reality, we only have some of the puzzle pieces right now. So in order to get a better idea of the big picture of CNS involvement, we performed bulk RNA sequencing on postmortem tissue from 9 DM1 patients on up to 11 brain regions. For further analysis, we focused on 5 brain regions, which we could compare to publicly available RNA sequencing data from the GTEx portal. What we found was that splicing dysregulation appears to be widespread across multiple brain regions in DM1. Across these 5 regions, we found there were 62 shared missplicing events, and many of these events are known to be misspliced in other tissues in DM1. For example, shown here, we have MAP2, MAPT exon 2, MBNL1 exon 5, and MBNL2 exon 5. However, one caveat of bulk RNA sequencing analysis is that we can't get an idea of splicing dysregulation in individual cell types. And this is particularly important in the CNS, where we have an extreme diversity of cell types present. So whereas in the past, when we're looking at muscle RNA sequencing data, we have just a few cell types present. However, in the CNS, this wide heterogeneity of cell types makes it really difficult to really parse out which cell types may be affected. So the questions we wanted to address was which cell types are the most affected, and is there a change in cell type composition? So in order to address this question, we performed single nuclei RNA-seq on postmortem frontal cortex that I mentioned was previously profiled by bulk RNA-seq. So far, we found there was dysregulation of gene expression in all cell types identified, with the greatest number of differentially expressed genes being found in microglia, and the greatest severity of gene expression dysregulation being found in endothelial cells. Interestingly, we also noticed there was an increase in microglial and endothelial cell composition in DM1. And interestingly, when we look at the correlation between the endothelial cell proportion and the splicing dysregulation score, which is a measure of average splicing severity within an individual, and these are the values obtained from the bulk RNA-seq analysis, you can see there does seem to be a correlation between the proportion of endothelial cells and the splicing dysregulation within an individual. So this suggests that endothelial cell proportion could be driving some of the splicing dysregulation that we see in the DM1 CNS. So we believe that bulk and single nuclei RNA-seq studies will help us to elucidate CNS involvement in DM1. We would like to develop methods for capturing splicing measurements in single cells so we can quantify splicing dysregulation on a single cell level. We would also like to identify transcriptomic dysregulation on a single cell level, as we believe this can help us to characterize distinct cell types and uncover how they may contribute to disease mechanisms of DM1. It'll be important to understand brain region and cell type specific dysregulation, specifically when thinking about developing therapeutics targeting the CNS, so we know whether we need to target all cell types or whether we should just target certain cell types that are affected. And with that, I'd like to thank everyone in the waiting lab, specifically those people underlined here who directly contributed to the project. Also, thank you to the Day Lab for donating these postmortem tissue samples, and thank you to our funding source. And with that, I'll take any questions.
Thank you for a great talk. Did you observe a tendency that some misregulated splicing would be more cell-specific, so some will be more associated with endothelial cell, more with microglia or other cell type?
So we haven't been able to look at splicing in single cell types yet. Currently, with current single nuclear, single cell RNA sequencing methods, it really just captures the 3' end of the transcript. And so in order to really look at splicing, we need to get better full transcript coverage. But we're currently thinking about ways that we can modify existing methods to be able to capture splicing measurements in single cells by single cell or single nuclear RNA sequencing. So we're working on developing those methods.
What's the lack of white matter? Is that because you didn't have that in the DM ones or in the control ones? Lack of interest.
Oh, yes. In terms of not having control samples for the other regions.
Your initial survey of 11 different tissues.
Yes. So we for the time being, we pulled publicly available control RNA sequencing data, but we're interested in getting control tissue for those other brain regions so that we can then prepare libraries and then have a more complete look at all those other regions.
So it was a gap in what was publicly available in the non-DM?
Yes.
All right, very good. Thank you very much. All right, next speaker is Curtis Nutter from the University of Florida in Gainesville from Maury Swanson's lab. This is poster number 23.
Thank you, everyone, for this opportunity. And today I'll be talking about how actually the choroid plexus and its missplicing can alter cerebral spinal fluid composition in myotonic dystrophy type 1, in addition to all of these other neuronal and non-neuronal types that we've heard about today. So as you all know, DM1 patients exhibit wide range of CNS symptoms. And as you can see, we're still trying to figure out where exactly that's coming from. What's interesting to me, especially, is the structural changes like the cerebral atrophy and the ventricle megaly. And one of the ways that we think this could be linked, all these wide changes, is by the choroid plexus and its effect on the cerebral spinal fluid composition. So to address this, you might be asking, why do we think the choroid plexus is highly affected in DM1, especially considering it's such a small tissue? Here you can see a mouse cerebellum alongside the hindbrain choroid plexus. There's a choroid plexus for each ventricle of the brain. And when we look at the DMPK RNA expression, comparing different brain regions and different regions of the body like muscle or even myotubes, we see that choroid plexus is really a standout through the whole body, even with higher expression shown here by qPCR for the fourth ventricle choroid plexus, even than the quad. So that's good, but does the splicopathy actually occur alongside the high DMPK levels, especially in our DM1 mouse models? And that is the case. We see accumulation of toxic RNA and splicopathy regulated by MBNL2. Here you see splicing of KIF13A exon 26 in both our 480 repeat knock-in mouse with changes in the heterozygous and the homozygous. And you can see that that is an even more profound change than the single knockout of MBNL2 on the right. So again, that's good, but does this happen in patients? And that's the big question. To answer that, we obtained post-mortem choroid plexus samples from control DM1 patients and Alzheimer's patients as a disease control. And we see quite prevalent choroid plexus missplicing, including in KIF13A exon 26, which I talked about earlier. And while this happens in DM1, it doesn't happen in Alzheimer's disease. So it's specific to the disease of interest we're studying. And again, trying to connect these molecular changes in the choroid plexus to physiological changes in the patients and these CNS symptoms we're also interested in. We wanted to see, can the changes we see in choroid plexus actually affect the composition of the CSF? So to do that, we chose one of the most highly altered splicing events in the choroid plexus, which is prosaposin exon 8, and looked in CSF patient samples, including 7 control and 10 patient DM1 samples, and looked for prosaposin protein levels, since the splicing event controls whether it's secreted or not into the CSF. And we see a dramatic drop in prosaposin CSF protein levels that correlates with the choroid plexus splicing that controls it. So that concludes what I'm going to talk about today. There's a lot more to talk about at my poster if you guys want to come see it or ask me on the app. But to conclude, we see prevalent choroid plexus splicopathy in multiple DM1 mouse models and patients. And this we're showing correlates with altered CSF composition. In the future, we'd like to directly link that to CNS symptoms, as well as identify biomarkers, which would be relevant to dysfunction of the choroid plexus CSF system. And with that, I'd like to acknowledge all the funding, the people who helped make this work possible, including many of you here, and the patients, of course, who donated samples. Thank you.
Question. Thanks. That's really exciting. I'm curious, tying it together with Emily's poster. I don't know anything about the choroid plexus. What's the?
They're endothelial cell types.
Okay,
yeah.
Thanks.
Yeah. Yeah, I would say, so the whole lining of the ventricle is endothelial cells, and they probably got the lining in the bulk RNA-seq, whereas the choroid plexus is a specialized endothelial cell that is hanging out into the ventricles basically and producing the CSF. So there's a relation between all those endothelial cells, but these are specialized, and we find they have a lot more DMPK and a lot more RNA foci.
Great, thank you.
All right, thank you very much. All right, our next speaker is Subda Mishra from the RNA Institute at State University of New York in Albany with Andy Berglund's group. This is poster number 34. You first. Thank you.
Can I start? So, hello everyone. This is my very first talk at MDF. So I'm a little nervous, but excited at the same time. So I'll be talking about one of my recent findings on quercetin, which we find as a promising candidate molecule for DM. So as most of us know, the transcription of the expanded repeat produces a toxic RNA that created a disease features the multiple mechanism, and the spliceopathy is one of the most explored one. And our lab and few other labs has established a proof of concept that reducing or blocking their transcription can rescue the downstream pathogenic mechanism. So, and to measure the level of this toxic RNA, one of my previous colleague Reddy had designed a DM1 helical line that help to that you can use to measure the level of toxic RNA versus no toxic RNA. This gene construct has a unique tag, so you can simultaneously measure their level. And this cell line has display the molecular feature of DM, which is formation of RNA foci and the missplicing. So I use this cell line and screen a natural compound library that has 390 compound, and I find that these top 6 hits below this red line, it's here's their name. They reduce the toxic RNA production by more than 50%. And here is their cellular toxicity, and centauridine is the least toxic among these hits. Here's the structure of centauridine, and centauridine is belong to flavonoid family, and flavonoid family has several different subclasses. And then further I pick 2-3 molecules from each subclasses to see which scaffold is best for reducing toxic RNA level. And we find that flavonol family is the best among all these subfamilies, and quercetin and centauridine has comparable activity. Here's their structural differences between centauridine and quercetin. Quercetin is present in most of the vegetables and fruits, so it's not toxic for human. So here is their cellular toxicity. It's better than centauridine. And we did a EU pulse labeling experiment to see if quercetin is reducing the transcript level through the blocking transcription or reducing its stability. So we took actinomycin D as a positive control to which previously shown to selectively reduce the toxic RNA level. So with the respect of time, its nascent RNA level is consistent. So it's reducing it's blocking its transcription rather than affecting its stability. And a dye displacement assay informed us that it's binding to the CTZ repeat. And this is molecular model just showing how this quercetin is fitting to the CTZ repeat. So next we test quercetin activity in DM1 and DM2 fibroblasts. So the leftmost psi levels for the healthy cells, then this is the disease one, and then increasing concentration of quercetin in the DM1 cells. So it rescued the missplicing of several missplice events, including ionosaur exon 11, and it reduces the DMPK and CNBP expression in DM1 and DM2 fibroblasts respectively. Then next we tested its activity in DM1 myotubes. So again, the same data format. this is high value in the control healthy cells, then this is in DM1 and then increasing concentration of quercetin. So at the 32 micromolar concentrations, quercetin significantly improved their missplicing. And at this concentration, approximately more than 90% cells were viable. So it's not causing much toxicity, and it reduces the DMPK expression selectively in the DM cells. So further, we tested the bioavailable form of quercetin, which is EMIQ, that has several glucose molecule attached to this location. So it's basically increased its solubility and its bioavailability. So we find that at 2 doses, 6-week and 12-week high dose concentration, there's a downregulation in the HSA transgene, but no change in the endogenous DMPK, which highlight that the activity of quercetin is repeat selective. And then after, it also rescue the missplicing of several events, including chloride and channel exon 7 event, and it also reduces the myotonia in DM1 mice. So the RNA-seq analysis informed us that it globally rescued the several missplicing event, inclusion or exclusion event, and it's not changing much of the transcriptome at the same time. So overall, quercetin is working is reducing the toxic RNA load in multiple DM models and rescuing the splicing, though its solubility is low. So currently, we're working on improving its solubility. So I'm developing a drug delivery vehicle so that we can encapsulate quercetin so that its solubility get increased and it better absorb in the intestine and then reaches selectively to the DM affected cells. And we have some promising preliminary data supporting our hypothesis. We also, to target multi-step the DM mechanism, we also are testing combination of quercetin with other known DM1 small molecule disease modifier. And we have preliminary data that shows it's have synergetic or additive splicing rescue. We hope to initiate its clinical trial pretty soon. My boss is working hard on that. And I'm pretty much thankful to all the Bugland lab member, Ready lab member. I'm thankful to all Masuki for doing all the mouse work and all the collaborator as well. My poster is 34. If you want to chat more about this, you can come to this poster, and my lab has several other posters, 26, 28, 29, 32, 36, and 54. So please stop by to our poster. Yeah, thank you.
Thank you.
I would like to welcome you, Professor.
One question. How about 2 questions?
Thank you. Thank you very much for the presentation. I had two small questions. The first one is, as you mentioned, you're working with high concentration of quercetin. So I was wondering about the plasmatic concentration in mice. Did you check if it's compatible with...
So if I understand correctly, you're asking its plasma concentration is in mice. So we're working on that thing. Yeah.
And my second question, it's more about the mode of action of quercetin. Do you have any clue about how it's working exactly?
Sorry, I can't hear you. Can you speak loudly?
Sorry, I was wondering about the mode of action, the mechanism of action.
So you're talking about mechanism of action. So we don't know exactly in our biophysical binding assay, it tells us it's binding to the repeat, but it's not selective binding. So we're still working on mechanism of action. Yeah.
Great. Thank you very much.
Thank you.
All right. And our last talk is from Lily Cisco from University of Rochester and John Lewick's lab. This is poster number 21.
Awesome. All right. Today I'll be discussing the benefit of amlodipine or ranolazine on a myotonic dystrophy bichannelopathy. So more muscle weakness and atrophy lead to approximately 60% of cases of morbidity and mortality in DM1. And in the Lewick lab, our goal is understanding the molecular underpinnings of this muscle weakness and muscle degeneration, as it's not well understood within DM1. We're also interested in how myotonia interplays with this as well. So we all know that hundreds of transcripts are aberrantly spliced in DM1, and this makes it very difficult to connect either single or small subgroups of these splice events to clinical manifestations. However, from work done in the Thornton lab, they've found a connection between aberrant splicing of 4 transcripts central to excitation, contraction coupling, and muscle weakness in the tibialis anterior of DM1 patients. And those transcripts are CAV1.1 with the DM splice defect of an exon 29 deletion, RYR1 with exon 70 deletion, CIRCA1 with exon 22 deletion, and CLC1, which has an inclusion of exon 7A, which leads to myotonia. That is a connection that has been made. Now, we are interested in studying these aberrant splice events beyond that complex splice background that's seen in the disease. So our lab took a reductionist approach and generated forced splice variant mouse models using CRISPR-Cas9 for CAV1.1, RYR1, and CIRCA1. And we're also taking advantage of an already available CLC1 null mouse model. We are studying these mice in individual of one another to understand them as single splice events and their impacts, but we've also taken these mouse models and bred them with one another to understand, essentially, is there a tipping point for disease phenotype? Now, of all the combinations that we've made, we've found a very striking result from the combination of that exon 29 deletion in CAV1.1 when combined with CLC1 loss. Now, what we see with this double trouble mice, as we call them, that exon 29 deletion with the myotonia, is these mice have significantly reduced survival, as seen in the red curves here. With these mice, they have an average life expectancy of only 9 weeks, and no mice has survived beyond 14 weeks of age. Now, I'll note that this is a reduction in survival when compared to just loss of CLC1 alone. Now, beyond this reduced survival that we're seeing, we also see reduced body weight in these double trouble mice, reduced in vitro muscle force generation in both hind limb and in diaphragm. They have altered respiratory function, and they have exacerbated myotonia both in vivo and in vitro. So this led us to asking, can we rescue these phenotypes with drugs that are either targeting CAV1.1 or targeting the myotonia? So to do so, we decided to treat our double trouble mice with drugs that are already FDA approved. To target CAV1.1 with that exon 29 deletion, we chose the calcium channel blocker amlodipine. And then to target myotonia, we used a known anti-myotonic ranolazine. We're essentially trying to treat each side of the coin. Now, we had hoped and expected, and we're still excited to see that in this yellow curve with the ranolazine treatment of our double trouble mice, this significantly improved survival in these mice. We were even more delighted and surprised and excited to see that in purple, amlodipine was also able to improve survival in our double trouble mice. And again, that's targeting CAV conductance. So beyond improving survival with both amlodipine or ranolazine, we saw that amlodipine and ranolazine were able to help improve that reduced body weight that we see in our double trouble mice. We also saw that both amlodipine and ranolazine were able to improve the exacerbated myotonia that we see both in vivo and in vitro. And ranolazine alone was able to help that reduction in force generation in hind limb and diaphragm, and it improves some of the altered respiratory function that we see in these mice. Now, I think the most striking of the experiments that we've done on these mice can be seen with time of writing reflex, where we take a mouse, we put it supine on its back, and we time how long it takes to write to all 4 paws back down on a level surface. Now, our untreated mouse, you can see, is really struggling to right itself. On average, at this age, around 10 weeks, it takes 30 plus seconds for this mouse to right itself. However, with daily treatment of amlodipine, we see that this mouse is able to write quite a bit faster compared to its untreated counterpart. At this age, these mice take between 5 and 6 seconds to write. And with the daily ranolazine treatment, we can see that these mice write even faster with an average of around 3 to 4 seconds at this age. So with what I've shown you today, I believe we've demonstrated that either targeting CAV 1.1 or targeting myotonia has therapeutic potential in DM1. Once again, my poster is 21 if you want to discuss this project further. And with that, I'd like to thank both the Lewick lab, my lab, as well as Charles Thornton and his lab at the University of Rochester. Of course, our funding sources for the lab, as well as my own. And finally, a special thanks to the Myotonic Dystrophy Foundation, both for their support through the Predoctoral Fellowship, as well as giving me the opportunity to share my work with you today. With that, I can take questions.
That's really dramatic. What is the cause of the early mortality in those double trouble mice? Is it cardiac? Is it they're losing weight and poor PO intake? Is it respiratory?
We believe that it's majority respiratory. So because CAV 1.1, that exon 29 deletion, the 4 splice variant is specific to skeletal muscle, we're not expecting any cardiac impact. But one thing we've noticed both from our, we do whole body plethysmography, as well as when we're doing our time of writing experiments, when these mice are on their back, they will sometimes essentially just stop breathing. So we believe that they're dying due to respiratory impact. Yeah.
Thanks. It's really a tremendous story. I'm curious, I don't know enough about amlodipine. Does it have any cardiac effects that might put it in risk of use clinically in myotonic dystrophy?
Yeah. Yes, amlodipine is typically used as a cardiac medication for high blood pressure, and but of some of the calcium channel blockers, we sort of chose this one for its being more safe than, say, other ones for use in DM patients with less concern. Yeah.
Thank you. It's a nice talk. So my question is, amlodipine is a 3rd generation calcium channel antagonist. And you said that we use it for hypertensive patient. So can amlodipine causes hypotension in this mouse model and also for DM patient? And how can we prevent this?
So we've actually done tail cuff blood pressure measurements for the dose that we're using for our treatment. And we saw no significant changes to either heart rate or blood pressure with the amlodipine treatment. So at least at the dose that we're currently using, we feel safe to continue on. Yeah.
All right. Thank you very much. So I don't know about you, but I'm feeling pretty good about the future of DM research. And again, reminder that poster session is at 12:30. So please be sure to check out all the posters. And thank you again. Thank all the speakers.